Hi, I'm Hayley Mattern, and I'm a senior at Old High School. My project was over the effects of UV intensity on mutation rates in Volvox carteri. In my project, I focused on the effects of UV exposure and adding to the Volvox mutant library. Volvox carteri is one of 50 known types of Volvox, which is a kind of multicellular green algae. It has approximately 2,000 cells and two cell types, germ and somatic. In nature, it can be found in ponds, puddles, and other bodies of still fresh water. There are several advantages in using Volvox carteri as a model system. It is easy to use, it has observable life stages, its small size, and germ soma differentiation. Volvox has been useful in researching the development and evolution of complex traits due to its simplicity. An example of this is inversion in Volvox and gastrulation in animals, as the similar processes of inversion can serve as a model for the more complex gastrulation process, and further our understanding of the physical processes involved. Another area Volvox are useful in is cellular differentiation. Learning how unicellular organisms can transition into multi-cellular organisms is an important topic in biological research, and the analysis of Volvox transcriptome of separated cell types is an important step in understanding the molecular toolbox underlying this development. Volvox is also a useful model for several areas of biological physics, such as hydrodynamic interactions and flagellar motility because of its ability to be grown in large numbers, the little variance in clonal cultures, its large enough size to be manipulated, and its simplistic structures. One of the current research goals is figuring out what genes are contributing to cytodifferentiation and cell development in Volvox. Through sequencing the mutants and comparing mutant sequencing results to wild type, we are able to narrow down what genes are contributors. Another benefit to this research was gaining information on mutations caused by UV exposure. As the ozone layer is damaged through global warming, there is more radiation exposure. Looking at the mutants' percentages as they relate to the intensity of exposure can provide more information about the types and amounts of mutations that can result. In my procedure, the Volvox were kept in tubes of standard Volvox media at a temperature between 25 to 32 degrees Celsius, with 16 hours of light and 8 hours of darkness. They are passaged every 3 to 4 days. Passaging is where a few Volvox are transferred to a new tube with fresh SVM when the nutrients run out in the past tube. In my project, 10 to 15 mother Volvox were transferred into a petri dish and then exposed to varying intensities of UV radiation, from 120,000 microjoules up to 150,000 with a UV stratolinker. Altogether, 50 Volvox were used separately for each intensity. The radiation method used was UV mutagenesis, which causes random mutations from pyrimidine dimers, which are molecular lesions. After exposure, the Volvox were then transferred into tubes wrapped in foil to prevent photoreactivation and to stabilize the mutations for 24 hours. The foil was then removed, and the Volvox were left alone for 48 hours under normal conditions to complete a life cycle. They were then examined under a microscope for mutants. The mutants were recorded for each intensity and then analyzed with a two-proportion z-test. In my results, I found two new mutants. The known Volvox mutants are shown in the Volvox mutant field guide. I found a new fuzzy type and another with odd gonadial cells. And with my z-test, all of the results p-values were above the alpha value of 0.05. This indicates that the intensity of UV exposure does not significantly affect mutation rates. In my project, it was found that radiation intensity increases, at least up to 30,000 microjoules, do not cause significant increases in mutants. The new Volvox mutants are currently being analyzed at the Donald Danforth Plant Science Center. Future directions for research could be a larger sample size, more intensities, or to use a different type of Volvox. I would like to thank Ms. Jennifer Hess and Dr. Gao for their help and support in this project. My works cited. Thank you for your time.